Hi everyone, welcome to Jitanda's Biology.
Today now we are going to know about cytoplasm. Cytoplasm. In this only meaning, cyto means what? Cell. Plasm means what? Liquid or fluid. Liquid or what? Fluid. Cytoplasm is the fluid or liquid present inside the cell is called as cytoplasm. For example, this is a cell now. This is a cell. And actually this is a cell means the outermost membrane of the cell is called what? Cell membrane. And inside we have this the daughter-like structures. The daughter-like structures inside this is nothing but called as a fluid. That is called as cytoplasm. That is called what? Nana? Cytoplasm. This daughter-like structure is called what? Cytoplasm. This cytoplasm is a liquid present here. See, that is around the cell membrane. Around the cell membrane, the liquid present, the fluid, or we can say the fluid present is called as cytoplasm. Fluid present inside the cell. Inside the water cell is called what? Cytoplasm. This cytoplasm, it gives the placement. It gives the what? Placement for cell organelles. Remaining all cell organelles are present in this cytoplasm itself. It may be nucleus, mitochondria, lysosomes, Golgi complex. This all the cell organelles we are going to know in the upcoming videos. So that all are present in this cytoplasm itself. And that is the duty of this cytoplasm. Next, coming into the protoplasm. Another liquid is called what? Protoplasm. Actually, it is not another liquid. Proto means nothing but life. Proto means what? Life. Plasm means what? Fluid. That is it is called a life fluid. It is called what? Life fluid. It is called as a life fluid. So, in the very ancient days, when that microscopes are not properly developed, only very simple microscopes has been developed and chemical investigation methods has been very in the beginning stage. At that time, the scientists believed that the essence of life, that is the origin of life, has been emerged out from this liquid itself. That's why it is called as a protoplasm. Why? Because the life has been formed from this liquid. So that's why proto means life. Plasm means a fluid. But after the development came in the science, the chemical investigation process maybe, or the equipment or instruments like microscopes has been very advanced. At that time, the scientist has been did so much research. Later they come to know that no, the life has been not emerged out and it is not having that much of significance like remaining cell organelles. So that's why later this protoplasm only called as a later it is called as what? The protoplasm. This protoplasm is called as is known as cytoplasm. This protoplasm is called as what? Cytoplasm in the latter. So in this way, we come to know about the cytoplasm as well as protoplasm. And these that is actually after the discovery of nucleus that we have been divided the cell nothing but nucleus and that is around the nucleus, inside the nucleus and around the nucleus. Why? Because nucleus only plays a prominent role in deciding, controlling all the functions of the cell. So that's it. The significance of protoplasm has been gone after discovery of nucleus and it is simply renamed as cytoplasm. Protoplasm is life fluid which is only given scope for the formation of life. But later it is a cytoplasm, just a liquid present inside a cell. Okay, of course it has its own function, but it is not that much of important and that much of key role in emerging of or origin of life from that. So that's why it is called as what? Cytoplasm. To know about another cell organelle that is called as endoplasmic reticulum. Endoplasmic reticulum. It is abbreviated as ER. Endoplasmic reticulum is abbreviated as what? Endoplasmic ER. See, these are the fine network of membranes. Fine network of what are membranes. For what purpose it is useful? It is useful for transport of substances. Transport of what are substances like proteins especially. Transport of substances like what? Proteins. That is the main duty of this. These endoplasmic reticulums, that is ER, has been classified to two types, that is SER and RER. SER and what? RER. SER means nothing but what? Smooth endoplasmic reticulum. Smooth ER. That is RER means R stands for rough endoplasmic reticulums. ER means endoplasmic reticulums. Why these rough endoplasmic reticulums are rough? Why? You see, these are rough endoplasmic reticulums, the dot-like structure, this area. And this area where there is no dot, that is called as smooth endoplasmic reticulums. Understood, children? Why this is rough endoplasmic reticulums? Rough means on their surface areas they consisting of ribosomes. They are attached with help of what are ribosomes. These small dot-like molecular structures called what? Ribosomes. What is the use of these ribosomes? These ribosomes are useful in protein synthesis. Useful in what? Protein synthesis. What by synthesis means? Manufacturing of proteins. Whereas smooth endoplasmic reticulum, there is no ribosomes, nothing is attached. So that's why their outer surface will be very soft. That's why they call smooth endoplasmic reticulums. So the smooth endoplasmic reticulums are useful in what? Lipid synthesis. They are useful in what? Lipid synthesis. Okay. They are useful in what? Lipid synthesis. So these are useful in the protein synthesis. These are useful in the lipid synthesis. Especially you take rough endoplasmic reticulums, they are useful of transporting the substances. Transport of what? Substances. Okay, from where? From the cytoplasm. From where, Nana? From the cytoplasm. From the various regions of cytoplasm. In the cell cytoplasm, you know, from the various regions of cytoplasm, they collect the proteins and they transport to the, that is cytoplasm to, that is various other organelles. To the other organelles. To the other organelles. That is the duty of this. And next you take, that is the main duty. And next you come to the, this moreover some of these biochemical reactions. Some of the biochemical reactions will also occur. Some of the biochemical reactions will also occur in the this rough endoplasmic reticulums. Some of the biochemical reactions will also occur in the rough endoplasmic reticulums. Biochemical reaction like respiration, something, okay, like oxidation, reduction. So like this, some process will be occurs in this. And next you take smooth endoplasmic reticulum, SER, which are present in the vertebrates. Which are present in what? Vertebrate. Okay, in the vertebrate animals, especially you take in the liver cells. They are present in what? Liver cells. They are useful for detoxifying. They are useful in what? Detoxifying. What? Detoxification of what? Toxins and drugs. Toxins and what? Drugs. That means detoxifying. What by detoxification? That means toxins means poisons. Sometimes when you take the drugs, that is medicines, at that time they will be stored in our body some residues. That will be gets that poisonous materials will get neutralized with the help of the smooth endoplasmic reticulum. Understood, Nana? So the smooth endoplasmic reticulums will detoxify the toxins as well as the drugs also. So this is about rough and smooth endoplasmic reticulums and overall about the endoplasmic reticulums. Next organelle is called as a Golgi apparatus. Golgi apparatus. And this Golgi apparatus are also called as a Golgi bodies. Or called what? Golgi bodies. Golgi apparatus are also called what? Golgi bodies. Camillo Golgi. Camillo Golgi is a scientist in the year of 1868. Okay, Camillo Golgi in the year of 1868. He has been discovered these the Golgi bodies. So in the respect of him or in the honor of him, these structures are named as, that is Golgi apparatus. As named as what? Golgi apparatus. from with the help of what you see? With the help of optical microscope. With the help of what? Optical microscope used to observe this Golgi apparatus. So these are the Golgi apparatus. And how this Golgi apparatus means? These Golgi apparatus are nothing but that is a membrane-like structures. And a membrane-like structures. They are having that is these membranes are forming the sac-like structures. You see, these membranes are forming what? Sac-like structures. Sac means what? A bag-like structures. In Telugu we call sanchi. So like that, a bag-like structures are being forming. And these in the sac-like structures, they are having the fluid-filled vesicles. Fluid-filled vesicles. That is a thin, small line, hair-like, a small, thin hair-like. That much thin hair-like structures are present inside this sac. They are called as vesicles. They are called what? Vesicles. And see, what is the duty of this Golgi apparatus? Means the Golgi apparatus are useful in a package of what? Proteins. Package of proteins before transport. Before what? Transport to the another areas. Before transport to what? Other areas in the or other regions in the cell. Okay, that is the main duty. See, whenever you go to the tour, what will happen? You have to pack up all the dresses. Okay, who will do that? Your mom will do that now. Same duty will be done by this Golgi apparatus also. They will collect all the proteins and all the remaining that is the materials which are being required at what place. That all will be get packaged and then it is get ready for the process of transportation. And next, this Golgi apparatus numbers. The numbers will be differs from one that is cell to another cell, and their numbers are more. They are more in numbers. These Golgi apparatus are more in numbers in the cells which produces either enzymes or else hormones. Either enzymes or what? Hormones. In that, these Golgi apparatus are more in number. And another thing is that when that is the proteins may be or the lipids may be the proteins as well as the lipids which has been produced by rough endoplasmic reticulum and smooth endoplasmic reticulum will be transferred to this that is the Golgi apparatus, and thereby especially you take proteins, they will get altered. They will be slightly altered. Altered means nothing but what? Changed their nature. And then after changing their nature, they are sending to the cell membrane. They are sending to what? That is altered, and they send to the what? Cell membrane. And they go to the cell membrane, and from the cell membrane they may be secreted out of the cell. Or if cell membrane only requires any maintenance, any repair is there on the cell membrane, then that cell membrane or that cell membrane will be get repair or else maintenance will be get repaired with the help of these proteins which has been transferred by the Golgi apparatus. Okay, now we are going to know about the activity 2. Activity 2. In that activity 2, we are going to know about that is mitochondria. You have to freshly prepare Janus green B solution. How much? That is 200 ml. For this you add 100 ml of water. 100 ml of water, and mix this both water 100 ml and Janus green B solution 200 ml, and then you keep it in a watch glass. You pour some amount of that liquid in the watch glass. Then you take onion peels, a thin onion peel in that. You keep this solution in that in the watch glass, and you keep this onion peel in this solution. And then you have to keep it for how much hours? Half an hour. Then you take out that peel outside. You have to take the peel out. Then you keep upon the slide. You keep it upon the what? Slide. Then you can observe under the microscope. You are able to observe under what? Microscope. Under the common microscope, that is a simple microscope, you are able to see the structure of mitochondria but not well clear structure of mitochondria. If you want to see the clear structure of mitochondria, like this mitochondria, you have to see with the help of that is a clear structure can be clear picture can be seen under what an electron microscope. With the help of what? Electron microscope. You take mitochondria is having of spherical. Its shapes are what? Spherical shape, oval shape, or it may be that is a cylindrical in shape, and a ribbon shaped, as well as that is a tennis racket shape, etc. In different shapes they are been present, and it is covered by how many membranes? Nana? 2 membranes. That is, it is a double layered membranes. It is covered by how many membranes? Nana? 2 membranes. So it is a double layered membranes. You see, Nana, this is the outer membrane, this is the inner membrane. Outer and inner. It is covered by how many membranes? Nana? 2 membranes. You take it is having a 0.5 micron, 0.5 micron width. The width is what? 0.5 microns width. And it is 0.2 to 0.5 microns, that is long. And it is 150 times smaller than nucleus. Smaller than what? Nucleus. Actually, nucleus itself very smaller one, but when compared to the nucleus, it is 150 times very smaller. Okay, and in each one cell, you can see nearly that is a 150 mitochondria. 150 mitochondria. Okay, nearly 150 mitochondria can be seen. And this is about mitochondria. You take in the mitochondria, in the inner membrane, the liquid is there. The liquid is called a matrix. That is called what? Matrix. And in the inner membrane, these foldings are there. These foldings are called what? Cristae. That is called what? Cristae. And these mitochondria are called as nothing but powerhouse of the cell. They are called what? Powerhouse of the cell. Why they are called as powerhouse of the cell means because the total energy required for the cell, cellular activities, has been that energy is given by mitochondria. That's why it is called as the powerhouse of the cell. Understood, Nana? And how they can give the energy? By participating in the cellular respiration. By participating in what? Cellular respiration. These mitochondria will be useful in production of energy. This is about mitochondria. Next, coming into the activity 3. Activity 3. This activity 3 is about to observation of chloroplast. Observation of what? Chloroplast or plastids. Plastids are the unique structures. They are present only in the plant cell, which are absent in the animal cell. And in this activity, same rheoleaf activity, you can repeat it. What you repeated in the beginning, and the same cell membrane while seeing the same activity, you repeat it. You are able to see the chloroplast. Or you can see another, by the help of another activity also we can see. That is with the help of activity 4. You can collect a small strands of algae, algae on the slide, and you can observe under the microscope. You can see that these are plastids. See, Nana, plastids are of 2 types. Plastids are of what? 2 types. They are divided into 2 types. They are chromoplast and leucoplast. Chromoplast and what? Leucoplast. See, Nana, leuco means white. Plast means nothing but plastids. Chromo means what? Colorful. Chromo means what? Colorful. So they are color plastids. So in that chromoplast, we are going to know about the chloroplast. We are going to know about what? Chloroplast. This chloroplast is present in the leaves. Present in what? Leaves. And the consisting of a special pigment called as chlorophyll. A special pigment called what? Chlorophyll. With the help of this special pigment, it is able to carry out the process of photosynthesis. That means they can prepare their own food particles by themselves by the process of what? Photosynthesis. So this is about the chloroplast. You take chloroplast, Nana. This is also double layered membrane, Nana. It is also covered by what, Nana? 2 membranes. It is also covered by what? 2 membranes. See, actually it is covered by 3 membranes, but here it is given 12. So outer membrane, inner membrane, and this is called a granum, and this colorless liquid, actually this white portion is there, that is called stroma, and this empty sac like cavity like structures is called a lumen, and these are called thylakoid membranes. They are called what? Thylakoid membranes. And you take these chloroplasts are different shape. They are ladder shaped, or they may be star shaped, or they may be spiral, or they may be that is etc. In different shape they are present. Ladder shaped, star shaped, spiral shaped, okay, and disc shaped. We can also see in the form of disc shape. In different shape they are being present in different organisms. And especially the main use of chloroplast is by containing of chlorophyll, they can trap the solar energy and carry out the process of water photosynthesis. Carry out the process of water na photosynthesis. Carry out the process of water photosynthesis. Going to know about one more cell organelle, that is called as lysosomes. Lysosomes. See, these lysosomes are spherical in structure. Spherical in structure. For example, this is a cell. Means like these small spherical structures inside the cytoplasm, that is called what? Lysosomes. These are what? Lysosomes. And these lysosomes are spherical in nature, and they have the destructive enzymes. Destructive enzymes they have. What the meaning of destructive enzymes means? Destructive enzymes means that enzymes which will be break down the total cell also. That much capability they have. So destructive cell. For example, if any materials which has to be get digested, that will go to the, whenever the materials has been, what materials? Actually the foreign materials, that is outside materials which are actually not unable to suitable, that is not suitable for the cell, that is disturbing the cell. At that time such a type of foreign materials will reach to the lysosomes. They reach to what? Lysosomes. Then what happen? These lysosomes will releases at that time these destructive enzymes. Releases what? Destructive enzymes. So thereby, while releasing these destructive enzymes, that enzymes what they will do? They will be, that is digest totally, that will burst, or we can say totally digest the cell, burst the cell. So that there would happen if cell only breaking, automatically the foreign materials also exited out from the cell. So that's why these lysosomes are called as the suicidal bags of the cells. Suicidal bags of the cells. Understood? They are called what? Suicidal bags of the cells. Who are they? Lysosomes. Next you come to the ribosomes. Next structures are called what? Ribosomes. These ribosomes are also, for example, you take a cell, inside the cell like this again small granular like structures present in the cytoplasm, they are called ribosomes. They are called what? Ribosomes. These ribosomes are present in the cytoplasm. There are two sides of these presence. They are present in what? They are present in the cytoplasm, or they are present on the endoplasmic reticulum. On the rough endoplasmic reticulum is RER. On the rough endoplasmic reticulums also they are been attached. That is the two sides are there for the presence of ribosomes. One is in the cytoplasm, next one is on the rough membrane of this rough endoplasmic reticulum. And what they will do? They are useful in the protein synthesis. They are useful in what? Protein synthesis. So they are useful in the protein synthesis. And coming into here, sometimes they are also present in single like this individually they will be present. Sometimes they are also present in groups. They are also present in what? Groups. Groups means like this a bunch, or else a number of all ribosomes will come and at one place only they are present. Then when they are present in the groups, they are called as polysomes. They are called what? Polysomes. Then they are individual, they are called as ribosomes. Okay, so this is lysosomes, this is about the ribosomes. By this we completed the total cell organelles, but we come to know that is about the cells. We have to know about now the cells. Whether the cells here in the that is the pictures, in the cells in the pictures they looks like a flat. They looks like what? Flat. But in reality they are flat? No, they are not flat. Cells will have the cells will have that is a height, next length, as well as breadth and thickness also. So these it looks to be flat in 2D dimension pictures, but when you see the 3D dimension pictures, okay, then you can see this length, breadth as well as thickness also. While you are seeing under the microscope also, if you focus, you change the focus of the lens. In high lens, you can see the thickness also. That means thickness is present means automatically we can say they are not flat at all. Okay. And later about that is a cell theory. If you come to the cell theory, there is a two important scientists. Who is that? They are Schilden and Schwann. Schilden and what? Schwann. See, this Schilden is nothing but a botanist. He was a botanist. That is a scientist in plants. Schwann is a zoologist. Is what a zoologist. And he was a scientist in the animals. So this the Schwann full name is Theodor Schwann. Theodor Schwann. Schilden is MJ. Okay, that is Schilden. MJ means Matthias Jakob Schilden. Matthias Jakob Schilden. These both person has been proposed the cell theory. These both persons proposed the first time what na cell theory. And actually before them also so many number of scientists has been knowing about the cells, but they are not conformly said about that events that is facts what they have been discovered about the cells in accurate manner. So first time they have been very daringly they said about so many facts about the plant cell as well as the animal cells. So that's why they got the credit of discovering the what cell theory. And along with these two scientists, another scientist also there, that is called as the Rudolf Virchow. Rudolf Virchow is one person who told about the cell division. Who told about what cell division. And his point is also these taken into consideration by the scientists of these two, the Schilden and Schwann. And the cell theory, that is salient features, that is nothing but the important features of cell theory. What are the main important features of cell theory means these are the important features of cell theory. First point is all the organisms are made up of cells. Made up of what? Cells. Of course, it may be a tiny organisms like ant to a great organism like a blue whale, elephant, or human being also. All are made up with the cells only. Second thing is the new cells. New cells are produced from pre-existing cells. Are produced from where nana? Pre-existing cells. What do you mean pre-existing cells means? Nothing but old cells itself. So pre-existing cells in the sense nothing but what nana? Old cells. Okay. So that is the new cells will not come from anywhere else. The old cell only divides and that gives rise to formation of what nana? New cells. That actually discovered by Rudolf Virchow. Okay. How means for example nana? This is one cell. This cell only breaks and forms what nana? Two cells. These two cells will breaks and forms what nana? Four cells. So like that all the new cells has been formed from the pre-existing cell. That is already existing cell from only the new cells are forming. So that is the second important silent feature of what? Cell theory.
Thanks for watching. Please like, share and subscribe.